It's fun to be here and meet a lot of you who I've Zoomed so much with over the last few years, and so many of you, it's been really a pleasure to see you in person. I'm going to talk today about our sequencing program at U of M and how we've integrated it with CBTN. U of M is sort of a recent entry to communicating with CBTN, but we really feel like we're taking off, and there's a lot of ways in which we're incorporating our program and our management of pediatric brain tumor patients with CBTN in a good way. So I'll start by just giving a background on our sequencing program. This is an ongoing program that's in both pediatrics and adults. It is a research study. So for our patients who have high grade, high risk or refractory tumors of any type, they're offered this sequencing program. It's a panel. It's a 1,700 gene panel. It is both DNA and RNA and germline. And this program has continued on mostly with grant funding, a lot of federal funding, and a lot of foundational and philanthropic support. So this is offered to patients, and it's CLIA certified, and patients get a report and their referring clinician within 2 to 4 weeks. So back in 2019, we actually compared this panel with, around the same time, all of these 4 centers published a paper about their experience with mostly high-grade glioma, but some with sort of gliomas of any types. And so we just compared the panels, and there were 177 genes that were across all 4, 148 genes that were across all 4 platforms. And we came out of that saying that a panel sequencing was appropriate for clinical grade for a report for real time for patients. I'll circle back to what we think might be a broader approach to sequencing, which may be better for a research perspective. So in 2020, we started communicating with CBTN. What had happened is a number of researchers at U of M had been part of those groups that had applied for access to data and samples. And so we really felt that it was in our, the right thing to do to deposit our sequencing data. And so what we did is we took our, what we call our retrospective cohort, and that is it has been deposited. The data has been deposited in CBTN. And the analysis of that data and a launch of a sort of separate project on Kids First, we think will be up by the end of this 2022. This will, through CBTN, make its way to the NCI through the CCDI. There's 2 prospective projects that are ongoing that involve this same sort of channel where sequencing occurs at U of M, sequencing data is then deposited in CBTN. So one of those prospective projects is PNOC 22 and 23. So these are both ongoing clinical trials that are at this point international and involve children with midline glioma. I'm not going to go too deep into what those studies are, other than as part of the consent process for those, you're offered MiOncoseq at those sites, and the consent process is built into the consent for these trials. This is funded by a number of the foundations who are very active in supporting CBTN. And then I'll circle back to this as well. This is a concept that's just about to open called the DIPG-DMG National Tumor Board. And this is something that was really spearheaded by Family Foundation with the goal of, for many of these patients where they're getting opinions from multiple centers with these high-risk tumors, to be able to get all the people in the room to help efficiency and to help the patients get a coordinated opinion. And again, we have received funding from the DIPG-DMG Research Funding Alliance to offer MiOncoseq to patients who are in need of sequencing through that tumor board. I'll talk a little bit more detail about each of these prospective projects. So PNOC 22 and 23 opened about a year ago and have really accrued quite quickly. As of when I put this slide together, there were 60 enrollments. And about, depending on the program, about half have done enrolled also in MiOncoseq. We request a frozen or FFP, like this sort of, the last talk was very timely for this. We really prefer, our sequencing team really prefers frozen. blocks and can work with a core or less than a core. Mostly what we've received is FFPE and mostly slides. And this gets back to the complexity of this patient population in particular, where tissue is absolutely hard to come by. So these patients often, by the time they enroll in these studies, have at most a core or part of a core left. And so our group has spent a lot of time figuring out how to get the most out of that small amount of tissue, often when it's 10 slides or maybe even less than 10 slides. And so far, the feasibility has been high. We, as was mentioned in the last talk, the FFPE slides are sometimes overfixed and there's issues with the library. And our group has generally tried to power through with this group to see if they can get a full sequencing report and sometimes even research sequencing. So I'll give you an example of that. This is one of the cases that enrolled PNOC, I forget which trial, but sequencing summary is here. And you see in the yellow here that this was slides and the tumor exome had a high PCR duplicate rate. So the group went ahead and was able to generate these sequencing report, which just based on the patient population, we would really trust these findings. And they make a comment about how the poorly resolved copy number, but you're able to see a copy number plot that we would trust. And so it's a situation where we're really able to, make really useful clinical information out of difficult cases to sequence. And that's one of the places where we think for this patient population, the current COG initiative is tricky because of the amount of sample that's required. The other thing that I'll say, and this sort of feeds into the next slides, is what we really want this sequencing to be able to do for us is to be able to get deposited into CBTN in real time. We're going to do this quarterly so that our collaborators who are working on all sorts of correlative and really progressive translational research related to the trial can get the data and do the analysis in real time. So as we're doing analysis on which patient cohorts are, how they're responding, we're going to be able to look at that sequencing data with the collaborators in real time. So in order to just dig into the weeds of what that might look like, I have to give you 1 or 2 slides of background on the drug that's the basis of both of those trials. And so many of you maybe have seen this are aware of this drug, but ONC201 is an oral drug that is complex in how it works, but is promising in these midline gliomas. It is CNS penetrant. And over the last 5 years, there's been a phase 1 that's now wrapping up that has shown some exciting early responses. This is just a snapshot of some of the clinical data that is currently being reviewed. Patients on the phase 1 who were treated at the phase 2 doing significantly better than historical controls, especially those with midline gliomas in the thalamus. So we worked with Sebastian Waszak, who you'll hear from later today, and who's active in looking at genomics within PNOC and CBTN data sets. And we were able to take the MiOncoseq cohort, patients who were treated at U of M, which was about 25 at the time we had this, who had sequencing, and look over the somatic calls and the RNA-seq, and then look at how did the patient's response. And so one of the things that Sebastian clued into was the percentage in reduction in size of the tumor, the maximal size and reduction, which is one of the metrics where we started to find some interesting correlations with different genetic findings. And if you look at the DNA level, you'll see that EGFR up on the top was 3 cases, and all those cases only had progression on treatment. We've since found this on about 6 cases where all of them have grown right through the drug. So it's an early signal to us, which we're still in the process of validating in the PNOC22 prospectively. And if you look down at MAPK, these are sort of a group of patients with different mutations in the MAPK signaling. All of those are patients with those alterations were much more likely to have that radiographic response. Again, we need to validate this prospectively. This is just a plot of the patients, the 4 or 5 patients who had MAP kinase alterations and their tumor area over time. And then here's the EGFR cases in the red and orange. And you see mostly growing or becoming metastatic on treatment. At the RNA-seq level, we see that mitochondrial gene sets were enriched in patients who had greater than the median reduction. in response to ONC201, and that sort of fits with the mechanism, which I didn't really get into, but that's our leading understanding of how this treatment is working. And again, at the RNA, EGFR and FOXG1, just at the RNA level, high expression was correlated with resistance to ONC201. And we've since validated this in larger data sets from tumor types and of all type, as well as glioma cases, where high amounts of EGFR or EGFR dependency correlates with resistance to this drug. So just with a small number of patients, we've got really exciting leads for how to predict who might respond to this drug. And so just knowing in real time that those are sort of our leading hypotheses about what might predict response, we now feed that back to our MiOncoseq team. And when clinicians are getting reports on patients who are being sequenced on this trial, they're also getting RNA profiles for these sort of leading candidate. And obviously we're not making treatment decisions based on these, but you're getting sort of this is that same patient who had the suboptimal, less than 10 slides. And in real time, they're going to get a readout of the EGFR compared to other tumors that have been sequenced, the FOXG1, which is downstream of EGFR, other markers that we think might predict response. And then when we get to a place where the clinical group is looking at the outcome for these early cohorts and whether various combinatorial studies with ONC201 are effective, we can also look at the paired sequencing data. And then getting a little bit further out into sort of novel ways of tracking response to treatment, one of the things that the people in my lab noted was that tumor cells that are these midline glioma cells that we treat with ONC201 have various response to ONC201. Some respond at a lower amount, some respond at a higher amount of ONC201. But one of the things that we saw was that these are two cell lines on the top that are relatively sensitive to ONC201. And what you see is very early on, ONC201 drops the mitochondrial total copy number of mitochondrial tumor DNA, even within hours of the treatment. And these two cell lines down here, which are relatively resistant, they really stay the same. We don't see that drop. So we think this might be a marker that we could look for in our patients, but then we had to sort of get creative of how we would know mitochondrial tumor DNA in the patient was coming from the tumor and not other cells, and how might you track that? And so Jack, who is a bioinformatician lab, had a couple of cool ideas. We started a cell line. This is a patient who was on ONC201, and he developed a cortical met, which you can't, it's not drawn out here. Oh, here it is. And our surgeons resected that, and we started a cell line in the lab. And what we've been doing for our cell lines and samples that we get from these patients with ONC201 is also just doing a panel or either whole genome or panel sequencing of the mitochondria. And indeed, this patient had a clonal mitochondrial variant, and that has held forward in the cell line. And what we do when we're looking at possible liquid markers of response to treatment that you might follow in plasma or CSF is a real easy way for us to model that is just media. Cells grow, and what they extrude into the media in cell cultures to us is pretty similar to actually the concentrations of tumor DNA and mitochondrial tumor DNA that we see in spinal fluid. And so indeed, for this cell line, we co-culture it with normal brain cells, and then we can look at what happens to the allele fraction of that patient-specific mitochondrial variant with response to radiation or with response to ONC201. And we think that if we can, at diagnosis in our patients, find patient-specific mitochondrial variants and copy number falling quickly as a marker of long-term response to ONC201, we could track those in the patient's spinal fluid, which we are collecting on these PNOC studies, or potentially even plasma. We worked with some experts in mitochondrial tumor DNA, and there was a lot of ways in which you can quantify the mitochondrial variants and copy number. But they all agreed that whole genome sequencing is really the gold standard. And even at a low copy number, you can get really high quantification of the mitochondrial tumor DNA. And this kind of fit also with other researchers who were feeding in on this sort of DMG group that whole genome was going to really offer us the best chance of discovery with this cohort. So what we did is we went back to the MiOncoseq team and said, okay, we had maybe 10 slides to start with, and we did a capture panel. Is there any way we could also do whole genome on that same data set? And they suggested that we go to the pre-library capture from the patient's germline blood sample and tumor sample, And sure enough, we've really been able to do whole genome sequencing. We're usually doing about 20x, but we think we could go more. That was honestly just limited by our budget. We're getting, in cases where we were able to generate a capture report, we've been able to get feasibility of whole genome as well, and also get really high coverage of the mitochondrial tumor DNA. I think it got mixed here, but 20x coverage gets you 10,000x coverage often in the mitochondrial tumor DNA. And I didn't mention these here, but these are some of the other things that Sebastian Waszak has been interested in getting paired germline and tumor sequencing on these cohorts. So we're excited about, and with such small samples, being able to capture a clinical panel, get that right back to the patients, but then on the research side, also capture whole genome. Now, I'll just mention quickly the National Tumor Board. This is spearheaded by Lisa Ward's team and the various foundations that work with her and her funding alliance. And then on the clinical side, Sabine Mueller and her team at UCSF has been leading getting this together, but we think this will open in November. And this is what our team would look like if we were on the Brady Bunch. You see us all with the colored backgrounds, and it's a good way, we think, for patients to be able to get opinions from leaders in sort of a one-shot effort. We'll have to see how it works. But this is going to be very integrated with CBTN, and not sure if it's getting discussed at any other time this weekend, but the data, both the sequencing data, but also the clinical data, and then the prospective data that these patients go through after being at the tumor board is going to be captured at CBTN. So we're pretty excited about that. And I think that's it. I just covered a lot. This is my lab and the collaborators that are working on the various projects we talked about. So I'll stop there. Thank you for your time.